Vamos a ahora dar la bienvenida a la doctora Alexandra María Moita Antunes, quien dará la presentación titulada Drug-induced adverse events, the role of drug bioactivation.
So, buenas noches desde Lisboa. So first of all, I would like to apologize because I'm going to speak in English, even though I completely understand the Spanish. My Spanish is awful. I speak a kind of Portuñol, so that's why I decided to speak to you in English. I would like also to thank the organizing committee for this invitation and for the opportunity of being here speaking about the role of drug bioactivation in the onset of drug-induced events, which constitutes one of my main topics of my research work. So, as you might know, adverse drug reactions constitute one of the rising causes of morbidity and mortality worldwide. It is indeed currently considered to be one of the 6 leading causes of death worldwide. And of course, it has a huge impact in terms of human health and healthcare costs with the escalation of these healthcare costs. So, it is being increasingly realized that most of these ADRs could be efficiently prevented by the adoption of pharmacovigilance measures. Indeed, for this, we need the development of new biomarkers, and we need to engage all the research community in order to provide further insights into the mechanisms involved in drug adverse reactions. One of the mechanisms that is more often involved in the onset of ADRs is the so-called bioactivation. So, as any other xenobiotic, when we take a drug, when the drug arrives to our organism, it undergoes a series of metabolic transformations. The ultimate goal of these metabolic transformations is to increase the water solubility of the drug in order to prevent its excretion. However, in the middle of these metabolic transformations, sometimes are also formed the so-called reactive metabolites. Reactive metabolites are most often electrophilic species that have the ability to react with biomarker molecules such as DNA or proteins, yielding covalent adducts. So once these adducts are formed, they will transform, or they will modulate the activity of proteins, and they can initiate allergic reactions or even initiate carcinogenic events. So reactive metabolites are widely accepted as having a key role in the onset of adverse drug reactions. And there are many examples of drugs that are currently in clinical use that are linked with ADRs through the formation of reactive metabolites and through the metabolic activation. In fact, the chemical structure of the drug or of its phase 1 metabolites is the primary factor ruling its propensity to be converted into a reactive metabolite or a toxic metabolite. And this is widely accepted within the pharmaceutical industry. And currently we know already that there are some chemical features or chemical structures that constitute a red alert in terms of toxicity. And every time that one of these molecules appears during the drug development process, they are subjected to some tests that aim to investigate if these molecules can be bioactivated to potential reactive metabolites. And the adoption of these criteria in really early stages of drug development have already decreased considerably the drug attrition rates due to toxicity during the drug development process. But these tests that are currently adopted are mainly focused on CYP-induced bioactivation tests, but less is known about non-CYP bioactivation or other metabolic pathways that lead to reactive metabolites. So one thing that makes difficult the identification of these kind of reactions is the idiosyncratic nature of certain ADRs. Idiosyncratic means that they are not dose dependent, they are not predictable. And this, of course, makes very difficult to reproduce in animals and during the clinical trials. So these kind of reactions are highly dependent on host factors. So these host factors are going to rule the predisposition to these kind of ADRs. And of course, this can only be monitored in post-market settings through the pharmacovigilance measures. However, and even though the key role of reactive metabolites is widely accepted, they are very difficult to monitor in vivo because due to their reactivity, they have a really short life in vivo. However, when they react with proteins or DNA, they yield stable covalent adducts that can be adequately identified and quantified. So these covalent adducts that can be monitored mainly by mass spectrometry-based methodologies can constitute surrogates of the reactive metabolites. And we can identify and quantify these adducts. So these adducts constitute tools that can be very valuable for the identification of risk factors or for the identification of the adequate doses to be used to each patient for the implementation of personalized medicine action. These, of course, will ultimately lead to the minimization of ADRs. So using these concepts and using the miniaturization of covalent adducts or adductomics tools, I have been involved in multiple studies, studying the bioactivation of multiple drugs, mainly anti-HIV drugs and anti-epileptic drugs. So here in this slide, I summarize some of the more studied drugs within my research group. And here are the faces of the students that were more involved in each of the projects. So one of the drugs that I have studied is abacavir. Abacavir is a nucleoside analog reverse transcriptase inhibitor used in HIV therapy. And abacavir is linked with acute hypersensitivity that have been demonstrated to be genetic related. However, it is also linked with long-term toxicity, mainly cardiotoxicity. And of course, this kind of toxicity raises concerns about the prolonged administration of this drug and warrants urgent and accurate risk-benefit estimations involving necessarily the development of new biomarkers of toxicity. So when we started studying this drug, it was known already that abacavir could be metabolized by alcohol dehydrogenases to halides metabolites. Unconjugated and the conjugated halide. So these for us constituted a red alert flag in terms of structure, because we know that halides can react with the amine residues of proteins, leading to the formation of covalent adducts. So but by this time, this was only an hypothesis. So we decided to investigate if this could indeed occur. So one of the most easy methods to monitor the formation of covalent adducts with proteins is to monitor the formation of covalent adducts with the N-terminal valine of hemoglobin. This is easy because there is a method, a very easy method, the so-called N-alkyl-Edman degradation, that allows the specific detachment of this kind of adducts, hesitant twins that can then be identified and quantified by mass spectrometry-based methodologies. So with this in mind, and since I am an organic chemist, I decided first to reproduce, or trying to reproduce this bioactivation and the formation of covalent adducts in vitro by synthetic methodologies. So this allowed us to prepare a synthetic standard of the dentwin expected to be obtained from the halides of abacavir. So once we had this synthetic standard, we started investigating if these adducts could be formed in vivo. For that, we have isolated hemoglobin from rats exposed to abacavir, and also we have isolated hemoglobin from 10 patients on abacavir therapy. So following Edman degradation of this protein, we were able to identify by mass spectrometry that this identwin could be formed in male and female rats, and also in all patients that we have monitored. So this constituted the first unequivocal evidence that indeed abacavir was bioactivated into reactive halides derivatives. And what was here interesting to notice, it was that only the conjugated halide is reactive metabolite that subsists long enough in men to react with biomacromolecules. So another drug that was studied within my group, it was nevirapine. Nevirapine is an anti-HIV drug, a NRTI that is the most used antiretroviral drug in developing countries where HIV is more prevalent. So even though the exposure to this drug is huge, it is associated with severe hepatotoxic and skin rash events. So when we started working in this project, there was already some hypotheses about the possible reactive metabolites that could be involved in these adverse drug reactions. So there was already the hypothesis that the phase 1 metabolite of nevirapine, 12-hydroxynevirapine, could be sulfonated to 12-sulfoxynevirapine via phase 2 mechanisms, or be transformed into quinone-methide metabolites through direct oxidation of 12-hydroxyneviripine or hydrolysis of 12-sulfoxyneviripine. So there was already the hypothesis that both these metabolites could react with proteins yielding covalent adducts. So once again, we tried to obtain some in vivo evidence for this potential bioactivation pathway. So to do this, we have used a very similar procedure to the one that I have just described for abacavir. So we tried to monitor the formation of covalent adducts derived from these two metabolites formed with the N-terminal valine of hemoglobins. So once again, we started by synthesizing the 12-hydroxy and preparing the synthetic standards of the identwin expected to be formed from these two reactive metabolites. So this allowed us to identify the formation of this type of covalent adducts in HIV patients on nevirapine therapy. So this constituted the first unequivocal evidence that indeed nevirapine could be bioactivated in vivo to reactive metabolites capable of modifying covalently proteins. However, both 12-sulfoxy and nevirapine quinone-methide give rise to exactly the same adduct. So with this experiment, we could not identify exactly which of these two metabolites was indeed the reactive one. So to investigate further this issue, we decided to perform incubations in 3D hepatocytes. So we decided to perform two separated incubations, one with nevirapine and the other with 12-hydroxynevirapine. We first investigated if 12-sulfoxynevirapine could be formed in these incubations, and indeed, in the nevirapine incubation, we detected a considerable amount of 12-sulfoxynevirapine. However, and surprisingly, in the 12-hydroxynevirapine incubation, no sulfoxynevirapine was identified. So this suggested that most probably nevirapine or any other of its metabolites could have a role in inducing the activity of sulfotransferases in order to produce the 12-sulfoxynevirapine. So once we had this result, so in nevirapine incubation, we had both the 12-sulfoxy and the quinone-methide, and in the 12-hydroxynevirapine incubations, we had the quinone-methide. So the next step was to investigate the distinct reactivities of these potential reactive metabolites towards different bionucleophiles. So we used N-acetylcysteine to mimic the glutathione conjugation, that is an expression pathway, and we used the bovine serial albumin to mimic the covalent modification of proteins. So, and we monitored the covalent adducts formed by high resolution mass spectrometry. So what was very interesting to observe it was that in 12-hydroxynevirapine, we obtained mainly the N-acetylcysteine adducts, which suggested that quinone-methide, even if quinone-methide can be formed, once formed, it can be right away detoxified by glutathione conjugation. In contrast, in the nevirapine incubations, we have detected mainly the bovine serial albumin adducts formed with histidine residues. So these suggested that indeed 12-sulfoxynoviripine should be or could be the reactive metabolite. So this is very important because this constitutes a metabolite that is only formed by phase 2 sulfonation. So this implies that phase 2 sulfonation can be metabolic pathways very important in the bioactivation of drugs. So these results made us realize that should be very important to have a quick test to test the sulfonation ability of each patient before being subjected to this kind of therapy. So very recently we have developed a simple method to measure the sulfonation in men using paracetamol as a probe drug. So this is potentially useful tool in precision medicine as it allows the study of individual variability in the response to drugs bioactivated by sulfotransferases. So another drug that we have studied is efavirenz. Efavirenz is also a non-nucleoside reverse transcriptase inhibitor used in HIV therapy. It is a drug of choice in developed countries, but it is associated with severe neurotoxic events. Efavirenz can be metabolized to two distinct phase 1 phenolic metabolites, 8-hydroxy and 7-hydroxy efavirenz, but only 8-hydroxy efavirenz is neurotoxic. In fact, this phase 1 metabolite is even more neurotoxic than efavirenz itself. So we have hypothesized that this could be due to the fact that this phenolic metabolite could be bioactivated to kinoid reactive metabolites. So to investigate these hypotheses, we have studied in vitro the reactivity of these two phenolic metabolites in biomimetic oxidative conditions. So, and it was very interesting to notice that indeed 8-hydroxy efavirenz could give rise to at least two distinct kinoid metabolites, but in contrast, 7-hydroxy efavirenz only gave rise to a quinolone metabolites that for which no toxicity is expected. So more recently, we have also demonstrated that these two kinoid metabolites can react with sulfur-based bionucleophiles, namely cysteine residues of proteins. So in our last work that is still in preparation, we were able to identify N-acetylcysteine covalent agates formed by 8-hydroxy efavirenz derived kinoid metabolites in the brain of rats exposed to efavirenz. So this demonstrates that indeed these bioactivation pathways can occur in vivo, and raises one additional question. Can efavirenz be bioactivated in the brain? So this is one question that we would like to address in the following our following studies. So etravirine is it was the first second generation non-nucleoside reverse transcriptase inhibitor to reach the market. Actually, efavirenz was developed to overcome the ADRs that are induced by efavirenz and neviripine. However, post-market reports on the severe skin rash and the hypersensitivity reactions have prompted the FDA to issue a safety alert on etravirine. So we tried to identify some potential bioactivation pathways of this drug. And in a first attempt, we have investigated these by incubating this drug in human liver S9 fraction. and we have monitored that was supplemented with glutathione, and we have monitored the formation of covalent adducts with this bionucleophile. So this allowed us to identify potential arene oxides that could be potential reactive metabolites. But the real interesting results were obtained in vivo when we analyzed urine of patients on etraveline therapy. So when we analyzed these urines, we were able to identify these glucuronide conjugates that were further conjugated with phenylalanine. So even though glucuronide metabolites are usually seen as excretion metabolites, these results suggest that they can be further bioactivated, and they can be conjugated with amino acids, namely proteins. So these not only suggest the occurrence of a new metabolic transformation that we evidence for the first time, but also raises questions about the potential toxicological implications of the bioactivation of glucuronide metabolites. One additional very interesting result was the identification of bis adducts formed with N-acetylcysteine. So these results have proved that indeed etraveline can be bioactivated in humans to bis electrophilic metabolites that are potentially capable of establishing cross-links with bionucleophiles such as protein-protein or protein-DNA. This is really a matter of recognized toxicological impact, since there is a considerable number of chemical genotoxicants that are known to establish intra- and intermolecular crosslinks with biomacromolecules such as DNA and proteins. So one last example that I would like to share with you is the example of carbamazepine. So carbamazepine is a first-generation aromatic antiepileptic drug that is linked with hypersensibility reactions, the induction of the autoimmune lupus, and also to some extent to pulmonary toxicity. The fact that this drug is administered in really high daily doses and the widespread chronic use of this drug by both adults and children warrants further investigations about the potential mechanisms that can be involved in the onset of these toxic events. So by the time that we started these projects, it was almost widely accepted that the potential reactive metabolite of carbamazepine, it was the epoxide, the carbamazepine epoxide that is formed by CYP bioactivation. So to investigate this, we wanted to see how this epoxide could react with bionucleophiles. So to do this, we tried to synthesize the carbamazepine epoxide. And the example that I will show you is an example how a terrible result in terms of organic chemistry can lead to very interesting results in terms of toxicity. Because indeed, my student failed in obtaining the pure epoxide, and she ended up with a mixture between that contained a large amount of carbamazepine and also our product. But since no reactivity between carbamazepine and N-acetylcysteine was expected, I told her, okay, go ahead with the reaction with N-acetylcysteine and we'll see what we obtain. So it was really puzzling for us the fact that when we monitor this reaction by HPLC with DAD detection, that instead of observing the consumption of the epoxides, carbamazepine was almost entirely consumed during this reaction. So these had shed some light for us that probably something very, very interesting could be involved here. So we decided to go further. And this time, Inês managed to synthesize pure carbamazepine epoxide. So we performed two separated reactions between the carbamazepine epoxides and N-acetylcysteine, and carbamazepine with the N-acetylcysteine. And it was very interesting for us to see that in both reaction mixtures, we could identify a similar adduct. But the yields of these adducts were much higher in the reaction of carbamazepine. So this was suggesting that carbamazepine itself, without the need of bioactivation, could be reacting with sulfur-based bionucleophiles, meaning that it could react with cysteine residues of proteins, for instance. So we decided to investigate these mechanisms further, these mechanisms of reaction between carbamazepine and sulfur-based nucleophiles. So, as I said before, we are organic chemists, so we performed an extensive study mechanistic study that allow us to realize that this reaction undergoes via the formation of radical that allows then the formation of all these products that were all characterized by high resolution mass spectrometry. So once we knew all about the reactivity of carbamazepine with sulfur-based nucleophiles, we decided to investigate if this kind of reactions could actually occur in vivo. So we decided to expose rats to carbamazepine, and then we have analyzed the extracts obtained from the livers of these rats. And using high-resolution mass spectrometry, we could identify some of these adducts that were already identified in vitro. So this really showed us that these radical-based reactions could occur in vivo. And that in addition to potential other enzymatic carbamazepine bioactivation, these non-enzymatic radical-based reactions may also be key players in the carbamazepine biotransformation, especially under oxidative stress and hyperoxia conditions. In fact, a clinical case of carbamazepine-induced interstitial pneumatitis was reported in a 19-year-old lung transplant recipient, which may, at least in part, be explained on the basis of the oxygen supplementation. So what we say here it is that the oxygen supplementation can be a risk factor on carbamazepine-induced pulmonary toxicity. So with these examples, I hope I have convinced you how important it is to engage the research community in studying further these mechanisms of drug bioactivation, as namely through the use of the atomics tools. If you have interest in this kind of studies, you might have the opportunity to submit some of your results in a research topic that I'm currently editing, on the Frontiers in Pharmacology, entitled Non-Cytochrome P450 Enzymes and Extrahepatic Drug Metabolisms, Challenges and Opportunities. So I would like to of course, to thank all the financial support, some of my colleagues that also participated in these studies that were not mentioned during the previous slides, and I will leave you with some slides of some pictures from Lisboa. So I would like to welcome you sometime in Lisbon. Thank you.
Muchísimas gracias a la doctora Alexandra.
De nada, de nada. These are the only words that I can say properly in Spanish. Spanish noches, de nada.
No, se entendió perfecto. Muchísimas gracias, doctora Alexandra. Me perdona, pero al inicio no pude hacer su introducción. Fue un error mío. Entonces, si me permite, voy a de igual forma hacer una pequeña introducción para usted, aunque está en el final, para todos nuestros asistentes. La doctora que acabo de presentar, la doctora Alexandra Antunes, es investigadora principal y profesora asistente invitada en el Instituto Superior Técnico de la Universidad de Lisboa, en Portugal. Ella es autora de más de 60 publicaciones revisadas por pares y fue asesora de 4 doctorados y 18 maestrías. En el 2014 recibió el premio CEFIC LRI Award, una de las subvenciones de investigación más grandes de Europa para toxicólogos de carrera temprana, y recientemente fue elegida como consejera del Grupo de Interés Especial de la Organización Hispana de Toxicología, basada en la capacidad de sintetizar estándares de biomarcadores y desarrollar metodologías analíticas sensibles. Su trabajo, como justamente han visto en la presentación, se ha centrado principalmente en los mecanismos de bioactivación, toxicidad de fármacos utilizados en terapias crónicas. Disculpa que esté hablando en español, pero obviamente era...
I understand. I completely understand. I understand the Spanish. I just cannot speak it.
Ok, perfecto. Muchas gracias. Ahora vamos a pasar a la sección de preguntas. Tenemos solamente un par de preguntas de la audiencia que nos está viendo a través del Facebook. La primera pregunta es de Alejandro Chávez Quispe, y es una pregunta que está en inglés. Good day. One question. What is the role of glutathione in the hepatic protection in case of paracetamol intoxication?
So in case of paracetamol intoxication, indeed glutathione plays a key role in detoxification. Okay, so people that have some deficiency in glutathione have more propensity in having some intoxication by glutathione. By paracetamol, sorry.
Muchas gracias. La segunda pregunta también es de Alejandro Chávez Quispe. Él pregunta, what is an adduct and why it is important in toxicology?
Mm-hmm. So an adduct is the product of a reaction, a covalent reaction between a reactive metabolite, a protein, or a DNA. So there is a formation of a covalent bond between the reactive metabolite and the DNA, so or the protein. When it happens in the DNA, the formation of these covalent adducts give rise to some errors that can initiate genotoxic events if they are not properly corrected. Okay, in proteins, for instance, they are going to modulate the protein function or by forming these covalent adducts that can be recognized by, they can be initiate allergenic events, because they are no more recognized as autochthonous proteins. So these are just some examples, of course. Actually, I am also not based on drugs, but based on chemical carcinogens. I'm currently investigating the role of the covalent modification of histones, for instance, in the onset of the carcinogenic events induced by the exposure to chemical carcinogens, for instance. So if an histone is modified, of course, we are modifying the post-translational patterns of the histone, or we are just modifying the conformation of histones, exposing more DNA, for instance. So the formation of covalent adducts can affect in very distinct pathways the toxicity of drugs and chemicals in general.
Muy bien, muchísimas gracias. Thank you very much. Muito obrigado.
Muito obrigada.
En todos los idiomas.
It was a pleasure for me.
Sí, muchísimas gracias, doctora Alexandra Antunes, por su presentación, por sus preguntas. Realmente es un gran gusto haberla tenido aquí con nosotros. Y la invitamos también sé que el horario es bastante complicado para usted.
It's already Saturday for me.
Entonces, muchísimas gracias aún más por ese gran esfuerzo que usted tiene para poder estar aquí con nosotros y con toda la audiencia que nos está acompañando por Facebook. Nuevamente, muchísimas gracias, doctora. Un gran placer tenerla aquí.
Thank you very much. It was a pleasure for me.